Yeah, I think we can get started. Right. Right.
Cool.
Right. Thanks everyone for joining in. It's my pleasure to have today Alexander Tong, who's a postdoctoral fellow at Mila with Yoshua Bengio, visiting researcher at Oxford with Michael Bronstein, and also a co-founder of Dreamfold, a protein design startup, and incoming assistant professor at Duke University starting July 25. Alex completed his PhD in computer science at Yale University in 2021 with Smita Krishnaswamy. His research interests span generative modeling, graph signal processing, and optimal transport to understand biological systems with a focus on cells and proteins. So it's a pleasure to have you with us, Alex. And with that, please take it away. Thanks.
Cool. And just one question, how long should this be, or what's the sort of, yeah,
Yeah, we can aim it for around like 45 minutes and like 15
cool.
minutes for questions and stuff from the audience. And yeah,
Sounds good.
sure.
Okay, great. And sorry, I have a small cough today, so bear with me. Yeah, so today I'm going to talk about sort of flow matching models and the applications I'm interested in, which are sort of cells and protein design and molecule design problems. So yeah, with that, taking it away. Okay, so flow matching is very cool. I would say, it started sort of last, beginning of 2023, maybe late 2022, and now has sort of been the default in sort of image generation and video generation and now protein generation. So sort of all these areas where you used to use just the diffusion model. I think oftentimes the math is much simpler with flow matching, and it just sort of works. So this is, Stable Diffusion 3, although there's been lots of advances since then. It's sort of a useful choice and very simple. And so in this talk, I'll sort of talk a little bit about it generally, what flow matching is useful for, and then go into some of the biological applications I'm interested in. And so in particular, right, image generation, very big, and I'll talk about sort of cell trajectories, protein design, and generating molecules. Okay, so all right, so a little bit of math, I guess, and why you might care. Okay, we've been using score matching since 2021, right? And the idea here is to use sort of the find your data distribution and noise it, and sort of this allows you to create models that iteratively refine and get better and better as you do inference to your sort of noise-free data distribution. This is great, but it turns out that sort of using the same type of math, you can get a much more general loss, which is sort of the flow matching. And the idea is to learn a vector field that is basically a mixture of conditional vector fields. And this can lead to sort of so the three things I like to emphasize is sort of faster training, faster inference, and also just easier to implement in general, because there's sort of less annoying math of Gaussians and sort of stochastic differential equations. And so this is a classic picture from Lippmann. This is depicting sort of the choice. If you look at that black dot as the data points, and sort of in the top right and in the bottom left is sort of a normal zero and Gaussian, then this sort of tells you the conditional paths to get to different places. So diffusion sort of moves in a very particular way because there's sort of it introduces a metric between Gaussians, whereas there's if you sort of move to more general flows, then you can learn sort of straighter paths, which are easier to learn. And so this is the idea is sort of this generalization is helpful in that you can make more choices in sort of paths you learn. And it sort of uses this general idea of it's actually easy to mix together vector fields to learn these models. Okay, well, that's a terrible looking picture, but okay, so let's see. The general idea is you're trying to learn these flows. So what is a flow? You're taking an initial point and you're pushing it along in time according to some vector field, and that sort of says both can push an individual point, but then you can look at also the push forward, which takes all of the sort of a distribution of points and gives you another distribution. So this is useful in generative modeling because you want to say go from a very simple distribution like a Gaussian to something very complicated like your data distribution. And so you want to learn these flows, particularly sort of the vector field that allows you to move, and then that will sort of applying these flows from a simple distribution to your data distribution allows you to generate new samples. And in images and in proteins and all these sort of things. Okay, so starting from the sort of the basics, right? Okay, so I would say normalizing flows is where this all started. The idea is very simple in that you can have a sort of a distribution that's very simple, a invertible transformation, and then sort of use trains of variables to calculate a loss. So this says basically the probability of your sort of target data is equal to where it maps to in the prior, and then sort of this change of variables. And so this allows you to train it using maximum likelihood. This is great, except the problem was that this is sort of very hard because you need very specific architectures so that they are actually invertible transformations. Okay, so yeah, and you can stack all these together and just stack a bunch of invertible transformations and get a deep one, and that's very useful. And that's what was done for a long time. The problem was that these would never sort of, it was very hard, right? Anytime you have an architectural constraint, they're much harder to train. And so this is really a sort of a big pitfall to these. And so we eventually moved to sort of models without architecture constraints, sort of like GANs and now diffusion models. And so, you know, one architecture that was very interesting was this idea of continuous normalizing flows. And that's the idea that, so while each individual transformation, you have to enforce its invertible directly by sort of architecture constraints is the most common way to do it. If you take the continuous time limit, one thing sort of interesting happens is that your individual transformations actually become invertible regardless if your network is or not, basically. So you use sort of a structure of an ODE to enforce that to enforce that there's an invertible map. And so that means your architecture no longer has to be invertible. So I thought this was very cool. And yeah, this is sort of a very interesting, you know, relationship. But these sort of were originally trained with maximum likelihood. So you can convert sort of the same ideas and calculate the maximum likelihood of your data distribution. So the loss will often be just like expected value of the negative log prop here. And so that's sort of just like maximizing likelihood of your data distribution. And so these were great, but of course I trained them for a while too, and a lot of other people did. But the problem was always like the simulation step could be very tedious, and so in particular, kind of calculating sort of this integral of the trace of the Jacobian was just very annoying, and sort of numerically unstable and time consuming. So while possible, these didn't train that well. And so yeah, this is sort of the basis for everything. Is we're actually flow matching models, and well, sort of diffusion is you can put it in this framework, but now we sort of have better ways to train these things. And yeah, so what does it look like, right? Yeah, we just have this mapping, and you're learning this flow. And so why is it invertible, right? Is because sort of each transformation along time, it's sort of a very small shift, diffuse motion measure of space. And so it's invertible by just sort of running the opposite vector field, the negative vector field. Okay, so now we the contingent normalizing flows were nice because you could train them with maximum likelihood. So you could, in theory, get any path that worked, right? Then your model could choose anything it wanted. But this is another way to think about it is, well, if you could pre-specify which way you wanted to go. So if you knew sort of your UT, right, your vector field that you wanted, then you could learn it by direct regression. And so this would be very nice because, well, now we have just then we have a regression-based objective. Of course, the problem is, so right, UT, if we just knew this vector field, that would be great. Of course, the problem is that we don't know this vector field. And so because, well, it's just computationally intractable to find a UT that maps from sort of your data distribution to normal distribution or vice versa. And so that would be great, except we can't do it. Okay, so that's where sort of conditional flow matching comes in, and really this trick that underlies all sort of diffusion and flow matching and sort of all these generative models now. And really, it's sort of the idea that you can model any distribution as sort of an infinite mixture of conditional distributions, small distributions that are very simple. And then I think of it sort of, how do you mix things together? It's sort of like summing over and mixing vector fields instead of problem distributions. Yeah, okay, so I was sort of alluding to this. Okay, so just a little bit about the main idea here. I think it's very important to sort of understand that, well, yeah, all the diffusion models and flow matching models are relying on this sort of very simple algebra. And really, this is the key part to all these methods. Okay, so a little bit of notation. Okay, so what does UT generating the PT mean? That means that sort of if you take your PT and apply UT to it, then you get sort of the next PT plus one, right? And so this is the basic idea is if you're, it sort of has to match in continuity. And so there's, you can go deeply into the sort of continuity equations here, but the idea is basically if some UT matches your PT and it is of this very specific form, then you can do this regression in a conditional way instead of a marginal way. And so when I talk about marginal, right, I mean sort of over everything. And when I talk about conditional, I mean sort of conditioned on something that I label here Z, right? But it could really be anything at all. But in practice, basically Z has come down to either be sort of a data point or a pair of data points or something like this is sort of most practical and often. So you can think about Z as sort of conditioning on a data point. So this, what does this sort of say? So the left-hand side is saying, right, if you did a loss, it was sort of, oh, I knew my vector field UT, I could regress my network to it at all positions if I also knew my PT of X. And that would be great. Of course, we can't do that. But what's much simpler is to sort of select a time, then sample of data points, and then sample sort of a noisy or some other distribution PT of sort of a position given my data point, and then regress to a conditional vector field, which generates that conditional probability distribution. And if these are much easier to calculate, then I can do this loss instead. And so this is sort of the idea is we can mix all these conditionals, whatever the conditional is, it's fine, as long as sort of UT follows this form. So as long as we're looking at sort of marginal vector fields that are mixtures of conditional vector fields in this very specific way, then everything is very happy. So yeah, this is the idea. Intuition on the math is just basically all you need to do is expand out both sides. So right, this is just expanding out the the norm, the squared norm, and you can see basically. Well, okay, you can't see my mouse maybe. Let's see if I can. Nope. Okay, can't see my mouse. But basically, the first term is the same. The last term doesn't depend on theta, and so it's sort of only this middle term that matters. Um, and when you plug in sort of all the expectations and times the middle terms, then you get what's in the previous equation. So that's sort of where the form of UT is derived. And so, right, what this says is basically when you take gradients with respect to the conditional loss, it's sort of equivalent to taking gradients with respect to the marginal loss in expectation. Yeah, so all of this is basically you're able to mix things together, and so now I'll give examples of sort of what things might you want to mix together. Okay, well, I can't click. Sorry, I've lost my ability to move. Hmm. Just trying to figure out what happened here. I opened the annotate and now I can't close it.
Thanks.
Let's see. Interesting. Um, why can't I? I can neither see my mouse nor change anything. This is a interesting problem. Ooh, this looks better. Okay. Okay, we're good. Okay, perfect. Okay, so what might you want to condition on? Okay, so we're talking about sort of a data diffusion up top here and a normal 0, 1 Gaussian at the bottom. Okay, so what might you want to do? one thing would be have Z be defined sort of as a data point. So sample your sort of Z's. Z's be equivalent to your X1s. So this means basically wherever you see Z, you're sampling a data point uniformly from your data set. And then you can define your PTs as sort of conditioned on a data point. Here's sort of your distribution over time. In this case, it's an interpolation between a small Gaussian around your data point and a normal 0, 1 Gaussian. And then we can look at sort of the mixture of all these things together. So the marginal PT will just be the sort of infinite mixture of all these Gaussians that are sort of interpolations between your data points and normal 0, 1 Gaussians. That's great. And the reason that we like Gaussians is because, well, once you're given sort of a sequence of Gaussians, it's very easy to also calculate the flow that matches those. And so you get a path of Gaussians, and it's very easy to calculate sort of the flow that gives you the next Gaussian. And so this sort of gives you an easy form for your PT of X given Z and your UT of X given Z. And that makes, in this case, basically everything is now tractable. I can do a loss that I can quickly sample data points, quickly sample sort of intermediate points that are PT of X given Z, and then calculate the flow there and do a regression. And so this is pretty much how everything works. There's sort of some variations to it. And yeah, so basically, yeah, when you mix them all together, then you get the correct UT. I think the most common one now is sort of this middle one, where you uniformly select sort of data from the initial distribution and the ending distribution. And this sort of gives you nice little tubes, and your vector fields are just like x1 minus x0. And so this is sort of the, I would say, the simplest form of thing. That seems to work quite well. And so this is what you would see in sort of the large scale video generation or image generation models. One thing that I was super interested in is sort of this optimal transport. So I was doing optimal transport flows for a while. One thing that's interesting is if you consider sort of Z that is not just independently pairs of X0s and X1s, but X0s and X1s that are sort of, there's some joint distribution. In particular, if your joint distribution is similar to an optimal transport matrix, then you can get very interesting flows, which are sort of like dynamic optimal transport. And the idea is to sort of move things to the closest parent X0 and X1. So yeah, that's the idea here. Is sort of the algorithm, right? Very simple. You select a time, you select sort of X0s and X1s, and then you can interpolate between them, selecting X to train your model, and then sort of do the simple regression, which is X1 minus X0, right? That's the conditional flow. So I would say this is a very simple version. And just what's nice is it basically just works almost all the time. And you can do it for anything. The OT version has one extra step, is just you do sort of well, this is the mini batch OT version. And so here you need to do sort of optimal transport between batches and then resample according to that OT matrix. And that'll give you sort of, instead of randomly paired things, right, and which you can see on the left, it sort of unscrambles things and makes close things go to close things. And so, yeah, we found this is very helpful in sort of improving training and inference. I would say this gives you sort of a small gain to anything you do if you're sort of interested in just unconditional generation. And so, yeah, you can see sort of the effect in 2D, right? I think in small dimensions it's very obvious. In higher dimensions, optimal transport is sort of it's an interesting quantity in high dimensions, but people don't really understand, like it sort of breaks down in some sense. It's very hard to estimate. It's unclear if and this is sort of a cursive dimensionality thing where sort of everything becomes equidistant as your dimensions get higher. So it's less clear what it means to do optimal transport if just everything is in high dimensions. Of course, if things are sort of on low dimensional manifolds, maybe it still makes sense. But yes, this is the idea. So yeah, using sort of optimal transport, we found in one of our works to say sort of unscrambling paths really helps just a little bit in sort of faster training. And you can see this sort of in CFAR results here. So just independent, which is blue, and OT, which is green, sort of slightly better FID. And in particular, especially when you sort of do the bottom, which is number of function evaluations during inference, it means the paths are straighter than if you did independent couplings. And so, yeah, that's sort of images. And right, there's lots of different choices, right? So now, what is the space that we play with, right? The space is basically trying to understand which conditional paths to pick, right? So I sort of talked about at the beginning where you did conditional flows, your network could pick the paths you want. Now the idea is to sort of pick what your conditional paths are going to be, so your PT of x given z, and sort of what your flows are going to be given that. Yeah. What is the form of the optimal map? Yes. Okay, so that's a good question. What is the form? So yeah, so the optimal map in our case, the one that most people consider is sort of optimal in the sense that it minimizes the average squared distance traveled. So between pairings, basically. So you sort of take L2 square distance between each pair of points according to matrix pi here, and that's sort of the optimal in some sense. So this is mini batch optimal in the sense that you sort of solve a optimal in each batch, and that works quite well. And actually, it works better than we understand that it should work. So I mean, in theory, you can like for some, if you have like small-ish data sets, so 50,000 or 100,000, it's possible to solve optimal transport between all of them. And in fact, if you do that, it often performs slightly worse than if you just did mini batch or even no batching. So there's sort of a very unclear relationship here. Actually, we don't really understand what you should do and what optimal means in this sort of learning case. The optimal transport sort of and dynamic flows are clear what that definition is, but it's not clear if that's sort of the best thing to learn when you're doing a learning problem between two distributions. So I think that's still one of the open problems is understanding better like what your choice should be of flows. Yeah. And so, right, practically most people have settled on the simplest choice, which scales quite well, but it's unclear, yeah, what to do and sort of yeah, like what should be optimal. And I think, in some sense, it's probably something in between, but we don't really know what that means at this point. And so, yeah, these are just 6 different choices of which flows you get. And you can see, like, even in 2D, you can get quite different things depending on sort of your choices for these different sort of distributions. And so, the OT is sort of picking a different choice for QT of given Z and straight path. And so, we summarize some of the choices, but I think from now there's sort of lots of other choices that you can make. Yeah, and so I would say most people settled on, right, this just the simplest sort of, I guess in this one, it's sort of rectified flow and what we call independent CFM. I would say this sort of sigma term matters a little bit in low dimensions, not so much when you do images. So it's sort of, yeah, but rectified flow, even with no rectification steps, is sort of the most useful in practice. Okay. Yeah, applications. So I've been interested in a couple different applications. I'll talk about sort of the application I've been working on a while and the one that sort of inspired this work on sort of optimal transport flow matching stuff. And so this is sort of a biologic problem, and I'll talk about sort of what the kind of interesting application is, and then how we could apply these sort of flow matching ideas to it. The second thing I'll talk about is sort of protein design. So how do you use these models in protein design applications, and in particular sort of what the weird protein manifolds are that you might use in that case. And then finally, I'll talk about sort of about molecule sampling. So this is something we're quite interested in, is sort of understanding dynamics and conformations of molecules, and sort of given instead of just given data, if you're given an energy distribution instead, then how do you sort of match that with the generative model? Okay, so I've been working on this problem for a while with Christine and Shabardi. Okay, so they're very interested in cancer, in particular triple negative breast cancer. And so in that system, there's sort of 2 different cell types, mesenchymal cells, which can float around in your body, epithelial cells, which are sort of fixed in place. And cancer sort of starts epithelial, does a transition to float around and can form metastases, and then settles back to do MET transition back into metastasize into new places. And sort of, understanding these transitions is very interesting to understand sort of metastasis and cancer. And so what they do is study these sort of tumor sphere models. So these are cells grown in dish in 3D, and you can see sort of the idea is to simulate this process of MET in particular. So transitioning from cells that can float around to stick in one place. And so, so, of course, the question here is like, what drives this transition? Because if you're able to understand what drives it, then you can maybe stop it with some drugs. And so the sort of system here is you're given sort of distributions over time. So this type of data is interesting because you can sort of sample the population, but it's hard to get sort of longitudinal measurements because every measurement is destructive. And so if you sample a cell once, then it sort of doesn't appear at the next time point. So you just get like slices of a population over time. And why is this the case? Yeah, so single cell is this interesting idea. It's very cheap, but you have to destroy things. You measure RNA, and then you basically get this matrix, which is like for each cell, what genes are present in that cell. And so it's sort of like a histogram of genes that are present in that cell at that current time and gives you a snapshot of what the cell is doing. So this little fun flow picture is the top part is a big bead, but every once in a while you can see a little tiny dot coming from the bottom left, and that's a cell. And so you sort of try to put a bead and a cell together and then measure the cell RNA for each cell. Okay, so how does this relate to flow matching and flows in general? Okay, so the picture is this, right? We have a bunch of different cell states over time, and we want to understand how the cells change over time. We get single cell sequencing data, and we get unpaired samples. But of course, we really want to learn the underlying trajectories, right? How we got to a specific state from those unpaired samples. So there's many possible pairings, and the question is which one should you pick, right? And the idea is, well, some sort of optimality. And so that's where this idea comes from, right, is to use sort of flows. So this is trajectory net. This actually uses pre-flow matching. This uses continuous normalizing flows to map over time and then sort of learn these trajectories of cells. So this is a nice method, but doesn't scale very well. Of course, now we can apply flow matching, right, to scale it to much higher dimensions and to provide much more fidelity. And so, yeah, some of the results I'll present on the biology are based off this earlier model, which is the one we could run a long time ago and have mice grow and lots of results. But yeah, we're working on sort of flow matching approaches to this, which I think are just much more stable and more useful in some sense. Of course, they also have this problem, right, in that you have to pre-specify which paths. So what's nice about the conditional normalizing flows, right, is you can learn these free-form paths. But of course, sort of we sacrifice something for this tractability in flow matching. Yeah. Yeah, so a couple of different ways to try this. I guess we've been interested in sort of manifold-based methods. And so that's what MioFlow is one of the extensions of this to do sort of in a latent space. But yeah, the idea is to sort of learn these flows over time for single cell and understand like how cells transition over time. And so why is this useful? Okay, so here's back to this triple negative breast cancer data set. So tumor spheres going from day 0 to day 30, and we want to understand the sort of transition and understand maybe what can disrupt it, basically. So now we look at this transition from epithelial, mesenchymal to epithelial, and we want to look at genes that sort of drive this transition. And so we can look at sort of what genes follow each other over time, and that gives you sort of ideas of possible causality. And then you could sort of narrow it down to sort of networks of genes that we know are related previously and sort of show up in our time analysis. And this gives us sort of upstream regulators of a gene of interest, which is CDH1, which controls this process. And these are sort of, we find in particular, sort of one of these genes, when you change it in the cells, it changes what is expressed. And so when you knock it down, which is sort of use CRISPR, or sorry, use siRNA knockouts to reduce it, then you get different expression. And then what's also interesting is, yeah, when you inhibit it, with a drug in mice, that actually you get less tumor volume. And so we think this is sort of an interesting application of these flow models to understand sort of first what gene they're regulating, which genes, and then knock it out and change it and see if that effect can help you in sort of these drug applications. And so here we use sort of trajectory net plus this greater causality networks to discover these new genes that we could potentially use as cancer treatments. Uh, cool. Yeah, so that's the application cells. Anyone have questions here before I move on to the protein stuff?
I had a quick question. So
Yeah,
you did
please.
mention you had like individual snapshots for the cell population. So like for flow matching case, did you like train like independence like flows, or like the flow was regressed for like each between T1 and T2 as well, or like just between
Yeah,
T0 and Tn? How did you
that's
include the information about T1 and T2 in the vector
yeah,
flow?
that's a great question. Yeah, so in general, what we do is so normally we say like Z is a sample, like a random sample of time zero and time one, and then you sort of draw a line between the two. In this sort of multi-sample case, then actually you draw a random sample from time zero, time one, time two, time three, time four, and then sort of draw a line between all of them, and then select a random point on that line. And so that it turns out is sort of equivalent. And basically, it's sort of saying, yeah, you could put any intermediate distributions, and all the regression stuff still works. And so that's sort of the algorithm we use here, just random selection. Although, of course, slightly better is to use optimal transport selection. So do pairwise optimal transport between each time point, and then select sort of the pairs that go together. And so, you can sort of play with the space. We're finding sort of, yeah, the OT pairs seem to work slightly better, but yeah, it's sort of unclear, less clear that, yeah, for this causality analysis, it's necessary.
Oh,
Yeah,
thanks.
cool. Okay, so yeah, so cells are cool, very hard. Proteins are interesting, and so we'll talk a little bit about that. Okay, so last year I was working on sort of these models called fold flow, and the idea is to sort of take our knowledge of these protein folding models and use them with flow matching. And so we sort of introduced the family of models, first in fold flow one, and then in fold flow two, that were generative models over protein backbones. So the idea is basically to generate from random noise valid proteins that you could actually make in the real world. Um, and so yeah. Okay, so just a little bit of background on proteins for those much less familiar. Uh, so the idea is proteins are a specific type of molecule. Um, and they're based on these building blocks of 20 amino acids. So you could think of them as sort of a sequence, like on the right, where you can just express as one of 20 letters, and they form these long chains, and then they form sort of some structure, some sort of secondary structure, which is sort of this intermediate structure. That's why you see sort of all these curly patterns and sort of flat back and forth sheets. And so the question, of course, is how do you represent these things, right? So we really are looking for, given a sequence, here's the structure. Of course, one of the problems here is that, well, there's not very much data on known structures. So in particular, there's like, well, less structures than we know sequences. And so that's why models like AlphaFold 2, which map, given a sequence, gives you sort of predicted structure, are very helpful. So that's called protein folding, because it sort of tells you, given a string, here's how it looks like, here's how it folds in space. But yeah, so these are the ideas. And so AlphaFold 2 uses these representations that are sort of like pairwise distances between all residues and 3D coordinates of each atom. And then I'll talk a little about what frame representation means in a second. Okay, protein backbones, what are they? So the idea is that you picture this long string, and you should picture it as sort of a long string of beads, basically. So each bead has a position and then orientation. So something like, it has a position, but then something hanging off of it. And so that's how I think about sort of proteins, is this long string of beads and with some direction. And so that's this frame representation. Is the idea is basically represent each location of each bead, and then sort of how it's rotated in 3D space. And so this is sort of a position and a rotation. And so that's why we call it sort of SE3 space, because it's a rotation plus this translation bit. And so, sort of complicated, well, different chemistry, but basically there's sort of four backbone atoms that we're looking at in space, and we sort of center at one of them, the C alpha atom. And so this is sort of the internal representation of almost all the protein models, is to look at these, or the protein only models is basically to look at these frames, a sequence of frames. And so you can, why is it sort of, there's sort of many representations. Why is this a good one? Well, I think it's because there's sort of some distances that curve, which is why these sort of relative orientations are useful. And then you sort of want it to be as independent as possible. And so sort of having all these independent pieces floating around is sort of nice to easier to learn. Yeah, and so this is the basic idea. Sort of the landscape of these models is that you have sort of different inputs and different outputs. So you can go from sequence to structure, which is called folding. You can go from structure to sequence, which is called inverse folding. Structure to structure, right? So there are all these sort of different options. And in particular, fold flow is sort of going structure to structure, and then fold flow 2 is going sequence and structure to structure. And so these are the basic ideas as like what sort of generative model are you using. This narrows down sort of the scope of our stuff here. So the main thing that was interesting is basically how do you do these flows over this interesting manifold space, which is the space of translations and rotations. And so initially, people were interested in sort of diffusion models over this space. And the idea here is to use the sort of analog of Gaussian noise in rotation space, which is IGZO, so isotropic Gaussian noise. And isotropic Gaussian noise has this form, and it's quite, quite a bit uglier than the standard Gaussian. And so this is one of the very nice things about flows is basically you don't have to think about how heat diffuses anymore. Instead, you can just sort of pick 2 points and draw a line in between them. And so this is why flow matching models is sort of much easier to do in sort of the space, is that you only have to look at geodesics or sort of pass between points rather than figuring out how heat diffuses, because heat diffusion on a manifold is very hard. So this is actually one of the simplest manifolds, and actually we have sort of a nice, easy parameterization of what heat might look like, but in many manifolds, it's actually impossible to do, basically. So this is why I think flow is sort of, if you're doing manifold things, it's sort of the right approach for diffusion. Okay, so let's see. Yeah, so this is just basically how do you evaluate these models? So on top, we sort of have our model that we're interested in. We go from noise to sort of a structure. Okay, so but how do you make sure that structure is good? So obviously the best way would be to make the sequence that corresponds to it in the lab and say, does it do what you want it to do? Of course, that's very expensive. And so most of the time people don't do this. This is the sort of computational way of looking at it. So you take your structure that you generate, you apply a different model, which gives you back a sequence for it or a bunch of them, and then apply another model with like root mean or something like this. Why is it a good idea? Is it's basically like a very strong filter of saying, I believe that this structure is something that can be realized because, well, 2 other models say it's something that they've kind of seen before and can make something useful of it. And so that's the basic idea of these validation metrics, is to say these are valid proteins because other models say they're good. Of course, that's sort of, there's many problems with that, right? If the other models are not perfect, which they are not. Yeah. See, I guess the type of analysis we do, and yes, the numbers are bold, which is nice. But the basic idea is you want to design things that are, realizable and new and diverse. And I think this is still a challenging problem, is to figure out, how do you design models that are not only sort of reflective of your data, but reflective of new things, for instance. Because obviously we don't really care about designing a protein that we already know of. We would like to design something new. Yeah. So one of the interesting things that we experimented with here is sort of this idea of zero-shot confirmation generation. So one of the things is that, well, proteins aren't static, right? So I showed this picture before, and this is sort of one structure. Given a sequence, there's basically, the structure moves a bunch, right? So even, just like existing, it moves around. When other things are introduced, maybe it moves around more, right? But the idea is that the structure is dynamic, right? And so, of course, we know a little bit about this dynamics, and so for some proteins know about it. And so here we sort of trained on data that was this known dynamics. And so this is sort of the idea is, yeah, you would like to say, instead of just sample one structure, have a generative model given a sequence sort of generates the all conformations, right? So that would be cool. So this is sort of saying, okay, we train up on a bunch of conformations here for given a new sequence, what does conformation look like? And so this is the idea of the task. I think FunFlow 2 does, a very okay job at it, not very good. I would say you could do much better. And so I think this is a really interesting direction to understand is how do you make generative models that understand dynamics? Because most of our data is sort of very static, but there's some data that is in terms of dynamics. And the other problem we'll mention is structure diversity. So one interesting thing people have sort of found, and we found too, is that these models prefer certain types of structures. And in particular, if you look back at this picture, basically, for short proteins, for instance, RF fusion really likes these alpha helices structures. And we don't really know why, right? But in general, most models prefer these types of structures. They're very stable. We can guess, right? But they're very nice and easy and sort of locally consistent, so maybe easier to generate. But it's sort of hard to get models to generate diversity in this sort of type of structure. So these plots sort of measure composition of different types of structure, and oftentimes it's basically hard to get to cover the full data space. If you look at all the models, they sort of like the bottom right rather than what the data looks like covering the whole space. So yeah, that's still a problem with these type of models, I would say. Although people are definitely looking at this. Yeah, so okay, just summarizing flows for protein backbones. The reason that we use flows is sort of if you're doing this frame space, it's much easier than diffusion. And in fact is also computationally easier too. And then it sort of we found it sort of gives you faster training and faster inference. So we compared that for sort of the same model architecture. So yeah, conditioning on sequence is very interesting. That's what FullFlow 2 does. And then understanding how to control these models, I think, is one of the next steps. And in particular, sort of how to control them so that they generate all the full distribution of structures given the sequence, not just one. Yes. Okay. So cool. That's protein design. Does anyone have questions on this before I move on to the last little piece?
Just
Yeah.
one quick question.
Yeah.
So when you have these uh, sequences and the corresponding structures, I think you would usually not know intermediate structures, right? It's just you have a sequence and you directly have the final structure corresponding to that sequence. Is that kind of usually the case?
Correct. Yeah. So that in a sense because you only have this final structure frozen basically, and so that we can measure it with
Okay.
crystallography. So yeah.
And then is it also the case that you will have like one sequence, but then many different structures that were synthesized or resulted from that sequence, and then you're just sort of trying to use the flow matching to make sure that you cover the space of all possible structures? Is that the idea?
Yeah, so that's the idea behind sort of this confirmation generation. So here you're sort of given data where you have one sequence and a bunch of structures for it, and given another sequence, you want to generate the structures for it, right? And so yeah, but most of these structures are sort of they're not measured experimentally; they're simulated, and so the measured structures it's sort of very rare to see many different conformations because it's so expensive, right? Because our data looks like sort of many more sequences than structures. Yeah.
Yeah, thank you. Makes sense.
Um, okay, cool. So last piece is molecule sampling. Okay, so talked about sort of this dynamics. I think dynamics is very interesting. And so this is sort of our attempts at a smaller scale to understand how do we learn models for this dynamics. Okay, so this is work that we worked on, I guess, earlier this year with people at Oxford. And the idea is, it's called scalable equilibrium sampling of sequential Boltzmann generators. The idea is to sort of scale Boltzmann generators and sort of tackle one of these problems. Okay, so what is the problem that we're tackling? What does equilibrium sampling mean? Okay, so normally, right, we're saying we're giving a data set and we want to match that data set. But in equilibrium sampling, we're given an energy and sort of that which defines a target distribution. And we want to sample from that, but we're not given data, right? So normally we're given data IID from new target and just want to generate that. But the assumption here is that we don't have unbiased data from new target, but we still want to sample from it. And so this is a much harder task. Sampling, I would say, is very unsolved, and it's much smaller scale. But I think it'll be very powerful if we can do it. And so this is one approach. So given a known energy function, you don't have the partition function, you want to sample from this thing. Why is this interesting? Well, oftentimes you're given a biased data set, and you want to find something new. But you don't know how to get from there to there. And so, one of the ideas is to sort of learn the sampling and sort of find these new modes and maybe the new drug that you may want. And so, one of the ways to tackle this is molecular dynamics, which basically simulates things, things moving around. The problem with this is that it takes a long time to go between modes, so it may be very computation expensive. And so what we want to design is basically something that sort of samples from the distribution just from the top. So from Gaussian noise, samples the correct distribution, rather than sort of being correlated over time and trying to make these paths and move between modes. Okay, summarized here basically. But modes are sort of hard to reach in molecular dynamics. So there's a big problem in molecular dynamics, and they're very interested in it, but still needs a lot of work. And so we're sort of tackling it from a different approach. How do you use generative modeling to generate all the modes at once, rather than just sort of these chains? Okay, so what is the way we're going to look at this now is Boltzmann generators. So the idea is to look at a Gaussian distribution and then sort of have some data. And so you want to generate that data, but then you want to reweight with sort of importance sampling, and that'll give you sort of your true Boltzmann generation. So generate your data and then reweight. Let's see. Oh yeah, I think I'm running out of time. So maybe we want to, let's see, I guess, okay, basically, don't have time for this, but what we were interested in sort of generative models with likelihoods, because you need to sort of do this reweighting step. And we find that sort of doing a sequential reweighting step at the end really helps. And one thing that we found interesting was that, so I've been looking at flow matching for a long time. Turns out in this case, actually, normalizing flows rather than flow matching work better. So using sort of the latest and greatest in normalizing flows, we found to work better than sort of these flow matching models that I've been working on for a long time. You can read the paper exactly why, but essentially the summary is because flow matching gets very slow to get likelihoods. And so normalizing flows, if you need likelihoods, are sort of more effective. And so the current state of the art was basically on this dipeptide, so 2 peptides, which is, very small relative to things I was just showing with proteins, but that was sort of the best we could do. And so 2 measurements here is basically energy measurement sort of measures local errors. Torus Wasserstein measures global errors in some sense. And so this was our sort of different metrics. And then we tried on much bigger peptides, so from 3 to sort of 6. And we found that, this sort of sequential Boltzmann generator way of doing things is much better. So I didn't tell you exactly what we're doing, but we think basically these ideas are very useful for sort of scaling the idea of sampling from a target distribution. Cool. Okay. So I will end there. Thank you very much. And happy to answer any questions or the discussion. Thanks.
Thank you, Alex. It's really interesting talk. Thank you so much. Let's open the floor to any more questions. Like if anyone in the room or online they have any questions, just you can unmute yourselves or just type it in the chat.
So there was a question in the chat in between, which says, how do you enforce constraints to make sure that the protein folding structures that you get from the forward mapping are like valid structures?
So this is actually a very interesting question and actually relates to what we're doing here. So basically, okay, so when I talked to Leon, who sort of is doing a lot of this work, basically he said that the Boltzmann generator strategy is great, but it doesn't scale because this reweighting requires sort of your generated samples to be like high fidelity and valid in some sense, right? Because if they're not valid, then they'll have terrible energy and you'll have to throw them away. And so to answer the question specifically is basically we currently generative models do nothing to make sure they're valid structures. And in fact, generative models are very good at sort of generating something that looks approximately correct, but is actually not valid in some sense, right? They're very bad at, in images, basically, it's very easy to learn a diffusion model that generates the global structure, nice things, but it's very hard to make them learn textures, right? And so local things is very hard for. And so actually what the sequential Boltzmann generator thing exploits is basically you want to use the generative model to learn the global structure and then use like refinement to figure out to make them valid structures. And so basically what we do is like gradient descent on the energy function is to make them valid structures. So you generate something that's not valid and then you refine it to make it sort of valid. And so that I think is an interesting strategy basically to make sure they're valid structures. Yeah, anyway, sorry, long answer to the question.
Thanks. Yeah, are there like any other questions in the room, online? Okay. Yeah, I had a quick question. So most of the times, like the Z is just like a pair of values. So you just take one X1 sample, one X naught sample. I was wondering, like, does it actually make sense to, work with like a batches or like model the kernel or something like that? Do you think that makes sense?
Yeah, yeah. So actually, we tried this in our in the OT conditional flow management paper we did, and so yeah, you could consider Z as batches of things, and all the math works better, and in theory, you get lower variance.
Mhm. Yeah. Sorry about that. Yeah. Just maybe wait a couple. Yeah. Hey, I was I think the connection's still
Okay, am I
bad.
back?
They didn't fix it.
Oh wait, just give me a second. Yeah, can you hear me?
Yep.
Yes.
Yeah.
Sorry, internet died here. Yes. So sorry, what were we saying?
Yeah, so typically you just like map one small Gaussian from base to one small Gaussian at target. I was just wondering, like, have you looked into like taking pairs or not just pairs between like base and target, but like pairs from the target as well and model the, I don't know, like kernel, you define some sort of a kernel and then maybe work with that, pair like two samples, like for example, from base, which are getting mapped to two samples from the target. Like, do you think that makes sense? Like working with like such small batches, like it might help with the training or like the sampling efficiency, because then you'll be able to get like a batch of samples at once or something
Yeah,
like that.
yeah, that's very interesting. Yeah, so um, so there's one paper on like Wasserstein flows or something by Dora, and I think these ideas of like generating groups of points at once and sort of not independently is very cool because, yeah, like, well, one thing is like the point cloud people, for instance, right? They don't ever generate one point at a time, right? Which is effectively what we're doing in some sense in many
Yeah.
cases. And so, yeah, like these ideas that yeah, you should sort of share information between your generated points is very useful. In fact, we've been looking at sort of methods that are sort of doing things that relate all the points that you're generating at once to make better generations. And so yeah, the sort of yeah, you should definitely, I think basically for controllability especially, you should look at pairs of points and things. But yeah, in general, it's a good idea. But just computationally harder, I guess, which is why people don't.
Yeah. Yeah, that makes sense. Right. Thank you so much. Yeah, are there like any other questions? Um, right. Yeah, if not, like let's thank the speaker one more time. And yeah, thank you so much, Alex. It was a really, really good talk. And the applications were really interesting. So thank you so much for taking out the time to give this talk.
Yeah, thanks. Thanks for having me.
Yeah, and yeah, we'll let you know when the recording's out. And yeah, thanks so much.
Perfect. Have a good one. See you.
Bye.